My name is Dr. Allie Gartland. I'm a reader in bone and cancer cell biology, and I work at the Mellenby Centre for Bone Research at the University of Sheffield. The problems we're trying to address in our research is trying to find out exactly why and how breast cancer spreads from the primary site to different sites in the body, and in particular into bone. We were trying to find out what it is that makes breast cancer cells actually metastasize, so move from the primary site and go into other sites in the body, in particular into bone. And what we did is we undertook several experiments. And the first one was using patient data, and we found that a particular type of condition called hypoxia, which is where there's low oxygen, seemed to be associated with relapse in these patients, and particularly with relapse to bone in ER-negative patients. So we looked at this in a bit more depth, and we took some, in the lab, we took some breast cancer cells. From this we then looked to see what proteins the breast cancer cells were secreting, and we found that there was a particular protein called lysyl oxidase, or LOX for short, that was highly expressed in the cells that actually go to the bone. So this was a significant finding because we'd identified this signal, if you like, that the cells give off when they have a tendency to go to bone. So we did another experiment whereby we looked to see what was happening in the bones of mice that had breast cancer, and the breast cancer they had high levels of this signal, this LOX. And what was happening is they were losing bone, and they were also getting very small holes in the cortex of the bone as well. And as I say, this is in the absence of any cancer cells. And then what was more significant was that these holes in the bone, these changes that were happening, we also found that the breast cancer had a higher tendency then to actually metastasize to the bone. So this signal was making changes in the bone that made it more preferential for the breast cancer cells to actually go into the bone. So we think it's a first step in what's making the bone a really nice place for breast cancer cells to metastasize to. And it's an important step because we know that this protein is released, and if we block it, we can actually prevent this metastasis to bone. So it could potentially be used as a predictor to identify patients who could then actually be going on and use adjuvant therapy such as bisphosphonates. Our work will help people affected by breast cancer in the future by identifying patients at risk of having bone metastasis early, and potentially as well by prolonging the quality of life by reducing and stopping the spread of breast cancer to bone. So we think that it will actually help breast cancer patients remain relapse-free for a lot longer. The ultimate goal of our research is to help those people affected by secondary breast cancer to live as long life as possible and as disease-free life as possible.